Okay, so to start off with our review on this stuff. So I want to kind of just walk through some stuff first. So with my other classes, I'm assuming that you guys don't have any specific questions that you want me to cover, right? So if you do, just chime in and let me know if there's something that you've been studying that you don't understand that you want me to cover. Just chime in and let me know. Otherwise, I'm going to kind of go through what I've done in some of my other classes. And we'll start off with HIV infection. So first things first, let's say you've had sexual contact with somebody that has HIV. And so if you are a female and you have sexual contact with a male, then the virus can be transmitted through the semen and then enter the body through abrasions that are created during sexual contact that we call micro tears, and then will enter the bloodstream that way. Whenever the virus first enters the bloodstream, it will be outside. Before it gets into any cells or infects any cells on the inside, it'll be an outside HIV virus. And then what will happen with that is, let's say that this is flowing in from that site of entry. And it's going to enter the lymphatic system and get filtered out through the lymphatic system first. You just have normal abrasions due to normal sexual activity. You'll still have some inflammatory response and stuff like that. And all that gets started off. We're going to filter everything back into the bloodstream first through the lymph, right? So we're coming back from the site of entry into the lymph, into a lymph node. And the first thing it's going to see is a B cell. On the surface of this B cell, we have a receptor that sees our HIV antigen. Stop. So it sees this. And that receptor that's on the B cell, we've heard talk about this before, but that's IgD. And then it's going to put that stuff that it picked up from outside on MHC2, because B cells can be antigen presenting cells. All right, and then the thing that's going to see this, we know MHC2 presents antigen, but MHC in general presents to what kind of cells? Do you guys remember? So yeah, CD4, so if we're talking about MHC2 will present to the CD4 positive ones, CD4 positive, and these are our T helper cells. And so this molecule here, coming off of this that recognizes MHC2 is CD4, whatever. And then these guys will go ahead and cross-activate one another. All right, and then this guy will produce TH2 cells as well as TH1 cells. All right, and the TH2 cells are the guys that can help promote this response and then lead to the antibody stuff. So once our B cells, because remember our TH2 cells, they are the captains of outside. Stuff. And then our TH1 cells are the captains of inside stuff. We haven't gotten to the inside part yet. We know HIV is going to get inside of a cell. We're going to get to that next. But for right now, it came from outside first before it got into a cell. And so our B cell saw it. It activated the CD4 positive T cell that also sees it. And so now our TH2 can go and activate all of our B cells, and our B cells will go through a process of proliferation and differentiation, always, making clones of themselves, making copies of the B cells out, making our memory cells, our regulator cells, and our plasma cells. This is important because the plasma cells are going to first make IgM that it'll secrete. And then this will class switch to IgG. And I'll say specifically IgG. We might also have IgA that will be apparent in the mucosa, but we probably won't have IgE, which is for our allergies or for our parasites, because we don't have those kinds of signals being sent out. So primarily IgG. This is going to be important to remember later on. So here's our IgG. All right, so that's great. That's the outside stuff. So let's talk about what happens on the inside, because we're talking about a virus, and that's what viruses do, right? They get inside cells. So what kind of cells does HIV infect? Does anybody remember? They infect CD4 positive helper T cells. Uh-oh. Right. So what happens is we have virus that was outside. This is always going to delete that. That's okay. That gets to be virus that's inside. All right. And so this might be a CD4 positive T cell that has, I don't know, it maybe has a T cell receptor that sees something else entirely. This might not be something that reacts to HIV. This could be literally any T cell that sees some kind of antigen of any kind that's being presented by an antigen presenting cell. And because this is an MHC2, this is always going to be, since it's CD4, so it's going to be MHC2. And MHC2 is only found on our antigen presenting cells. It doesn't matter what kind at this point, right? This is normally what this T cell would do. But now this T cell is infected with HIV. And HIV literally, if I were to take this, HIV literally binds onto a molecule on the surface of our CD4 T cells. That is CD4. So this binds onto here and then gets into the cell that way. It uses CD4 to do that. All right, so now we've got our virus on the inside of this cell. If this cell is infected on inside, but it's a CD4 positive T cell, right? Okay, so what did these guys have to do up here if we were looking at these CD4 positive T cells up here? When it got activated by our antigen presenting cell, which was a B cell in this case, when we turned into our Th2 and Th1s, we also had to go through that proliferation and differentiation step. What do we do whenever we're proliferating? We're making copies of the cell, right? In order to make copies of the cell, you have to have genetic information that has the information to copy the cell and make all of its parts and everything like that. So if that's the case, and these are eukaryotic cells, and they have to have genetic information in them, what part of the cell is that genetic information located in? Nucleus, right? So this is a nucleated cell. So all nucleated cells have the ability to get infected by viruses, and all nucleated cells have the ability to signal for help with MHC1. I know you guys have seen this before because I've even asked a question about it in a quiz or something like this. So these guys, even though this is their normal job is to have CD4 and react to stuff presented on MHC2, these guys infected on the inside with something have to be able to call out for help too. So they'll present that inside stuff on MHC1. So what's going to respond to that? A T cell, because it's being presented on MHC. But if it's MHC1, it's a different kind of T cell, isn't it? It's a CD8. This is our CD8 positive cytotoxic T cell. All right, you guys following? Check and make sure I'm not missing anything on this. It doesn't pop up with my chat over here. So I'll try to look back at these occasionally because it doesn't pop up on my thing. But okay, so all right, we're following along so far. All right, so our CD8 positive cytotoxic T cell, it can't do anything though without a signal from a TH. And it needs a Th1 cell to give it a signal for HIV specifically. So this might be a CD4 T cell that responds to just some kind of antigen. Maybe it responds to E. coli. Maybe it responds to flu. I don't know. It doesn't matter. But this CD4 T cell is now infected inside with HIV and is calling out for help with HIV antigen. This is a CD8 positive T cell that sees HIV antigen. So it needs to get a signal from a Th1 cell that also sees HIV, which we activated up here. Our captain for the inside stuff. We'll come over here and activate this. Now, our CD8 positive T cell will be able to make granzymes and perforins to kill our infected cell, which happens to be a CD4 positive T cell. And any CD4 positive T cell that gets infected with HIV will call out for help on MHC1, and then these guys can now go and kill them with their granzymes and their perforins. Now, HIV itself, having an HIV infection in your CD4 positive T cells doesn't cause them to die. They could live forever with no harm whatsoever, just with HIV in there and incorporated into their DNA. The problem is that the CD8 T cell response over here is going out and killing our infected inside guys. And they happen to be our CD4 positive T cells. So now suddenly all of your captains that respond to all of your infections are getting killed because they're infected with HIV by your CD8 T cells. So when you have HIV and your CD4 T cell count goes low, and then we said that you have AIDS, your CD4 T cell count is getting low because your cytotoxic T cells are killing them. So that's what's really going on with HIV. So that's the real problem there. So now are we starting to see how this is a little bit, getting a little more meta with this and how this is all relating back? Yeah. So I had a question from one of my students in my Wednesday class. So something else that helps us think in a little bit more meta of a thought process here. We can, if you have leukemia or something like that. So let's say you have leukemia, right? You have overgrowth of one of your white blood cells. So what do we do to treat somebody that has leukemia? Give them radiation therapy, right? And then replace their bone marrow. We'll kill all of their white blood cells, all of them, and then give them a transplant from somebody and give them new ones in the bone marrow, right? That's where your stem cells are for all of your white blood cells. So why can't you do that for HIV? You can, it turns out. It's just you have to get every single one of your HIV infected CD4 T cells killed with the irradiation. So there has been at least one case in 2023. There was a woman in Geneva, Switzerland, that they irradiated her and gave her a transplant with new bone marrow. So she got all new CD4 T cells. And as long as the virus was all gone from her body too, those new T cells don't get infected with HIV because the cells that had the HIV in them got killed. And so they were able to, through retroviral therapy, make sure that her viral load was 0 in her bloodstream, irradiate all of her white blood cells and replace them with new ones. And as long as there was no more HIV anywhere in her body, her new cells didn't get infected and she hasn't had any HIV since then. She's been cured. It actually worked. She was cured. I mean, there's reasons, yes. So there is a possible way. And now that hasn't worked for everybody because the irradiation therapy doesn't necessarily get rid of the virus. So if your, medications haven't actually got you down to a true 0 on your viral load, then that's the whole point is going to be moot. I mean, if you're giving somebody new white blood cells and it gets reinfected with HIV, that's the problem there. So that's one case where that has been effective. I don't know if there's more than one out there, but I saw that one for sure. And again, that's a very recent thing. That was 2023 that they did that. So the price and the risk, your insurance company is for sure not going to cover that, right? You, first of all, put yourself at risk. If you were infected with the HIV, then, should have had, things in place, prophylactic things in place to prevent you from getting the HIV. So your insurance company isn't probably going to pay for it. If you found a scientific study that's willing to pay for it, then you probably get lucky. because their funding is only going to be limited for that as well. So this person got lucky probably with that scientific study and all of that. So why would our, insurance, our medical, pharmaceutical companies, and everything like that not be pushing something like this more for some of our HIV infected patients? Well, probably, first of all, it's hard enough to find bone marrow donors for people that have things like leukemia and stuff like that, let alone trying to find the justification for a bone marrow transplant for a patient that has, a disease, an STI, basically, or from drug use or something like that. So there's that, but also then there's the other side of it, which I hate to bring up because it sounds conspiracy theory, but there is the other side that this is a fact that pharmaceutical companies would lose out on money on these drugs that are now very, very popular if there was suddenly a cure for this disease. And now it's not a cost-effective cure, but the pharmaceutical companies aren't making money off of that, So at the same time, there's that sort of an argument as well. So are there cures out there maybe for some of these diseases that, are easier to treat, just the signs and symptoms of, with our pharmaceutical drugs? And so that's why we lean on that. I think that's more of what the issue is, right? It's more cost effective for insurance companies, for the patients to pay for the medications, as opposed to trying to get, a whole transplant to replace something like that. So, I mean, it is what it is. Let me see if I'm missing anything off of this. I'm gonna double check all my stuff. Yeah. Okay. Okay, so we'll just make a note on here, saying that now our CD4 positive T cells are killed by our CD8 T cells. All right, so that's it for our immune response on the HIV side of things. But what about how the HIV virus actually hijacks our cells and replicates itself? So now we're coming to the life cycle of HIV. So let's say you have your CD4 positive T helper cell, and it's infected on, I want to make it way bigger than this. I don't know why I made it so small. Make it even bigger. Okay. And it's got, inside of this CD4, we've got a nucleus, and this is where our host genome is going to be. All right, cool. All right, so this is in our nucleus. This is our host genome. And first we have our virus, and it's out here. Let's draw it a little fancier than that, because it's got its nucleocapsid and it's got its own genome in here, and it's got an envelope, and that envelope has spikes on it. And don't we just love it? All right, so this guy comes and he attaches to a surface molecule up here, which happens to be CD4. And then this guy gets inside. When he gets inside, he'll release his genome, which is an RNA strand. That RNA strand will be able to come into the nucleus. And when it does, it'll go from RNA to double-stranded DNA. We go from RNA to DNA. And the enzyme that does this is reverse transcriptase. And then the DNA will get inserted into the host genome. Now, this is the provirus. It's inserted into that host genome. So that's great and everything. It's inserted in there, but how do we get the new virus particles out of here? So now what happens is that provirus will be able to make RNA particles from the HIV DNA that was in the host genome. This is the normal process of transcription. All right. And then this RNA, we can either bring it out here and get it ready for packaging, or I don't really like how I drew that. Hold on. So we'll have some RNA that'll come out. Well, that's fine. Sure. Come out here for packaging. I just feel like I'm drawing this way too big. RNA. All right. Or we'll have some that are going to go through this process down in here. Does this look familiar to anybody? What does this look like I'm drawing down in here? This is ribosomes, exactly, on the endoplasmic reticulum. So then some of these guys will go through these ribosomes and go through the process of translation to make more proteins. And then the proteins will move through the ER. And then they'll go through the Golgi. And then they will proteins. They'll move through the Golgi apparatus and then eventually get put on the surfaces of our cells. All right, so we're going to stud our whole surface of our cells with these spike proteins. Now we have our RNA. We also have our capsid proteins that can then come along and encase our RNA and then push its way out by budding. So then we will have RNA enclosed in a capsid. Remember, this structure right here is called the nucleocapsid. RNA plus capsid is the nucleocapsid. Okay. And then this guy now has budded through the membrane and he has the spikes all over him. Yeah. So we've gone through getting our outside virus into the cell through our CD4. We've gone through incorporating it into the genome through the reverse transcription. We also have the thing that incorporates into the genome is called the integrase. All right. We're making our proteins. One of the things that it does is it'll make one big old long protein and then clip it into the capsid and the spikes and the reverse transcriptase or whatever else is made. And when that clipping happens, that's going to be something that is done by an enzyme in the virus called protease. And so those can all be targets of antiretrovirals. So we can highlight those guys too. So here's a target, integrase. Here's a target, reverse transcriptase. And then whenever we're dealing with the proteins, we can target that with the protease of the virus. So those are all good targets for antiretrovirals in that stuff. All right, are we kind of following along a little bit? We're taking all of everything that we know about how the cell works, about how the viruses get in, as well as we talked about our immune response to that guy and why we killed off our CD4 T cells. All right. Now, let's say you have a patient that claims they're HIV positive. All right. So first things first, with HIV, if you're newly infected, you're not on any of your antiretrovirals yet. You're newly infected, okay? Then we can test for your virus in a couple of ways. Let's see how I approach this over here. All right, we might test for the virus genome. If we're testing for the virus genome, HIV has an RNA genome. So if we're testing for our genome, what's one very common like genetic test that we can do to see if something is there? PCR. PCR is basically making copies of specific DNA sequences. All right. So if we want to check to see if your patient has HIV genome in their system, then we will do something called reverse transcriptase PCR. And what this does is we will take your patient sample, let's say it's the patient blood, treat it with reverse transcriptase. Is Meatball here? Guys, come here, Meat. Come here, Meatball. Here he is, everybody. Oh my God. This is my husband, Brad. And this is Meatball. Are you gonna hang out?
Yeah.
Just sit in the audience and try to engage. This is Meatball. You guys who are online, you're missing out. He's like a little meaty guy here. Meaty guy. You gonna go hang out? Just make sure you don't pee anywhere. You wanna go say hi? You're not so sure? Go say hi. I'm not so sure. He might chill out in a little bit and come see you guys. He's friendly, he just is a little skittish. Yeah. Anyways, so all right, so reverse transcriptase, the same enzyme that HIV will be using to convert its RNA into DNA that then gets inserted into the host, we can actually hijack that in genetic engineering, buddy. And then, yeah, you're a good boy. And then use that to turn the RNA into DNA. So the patient blood, where we have the RNA of the HIV virus, we'll then use reverse transcriptase to turn it into DNA of the HIV. Where'd he go?
He's over here.
Here, buddy. Oh, there you are. You want to lay down?
There's a reason why he's called Meatball.
Yeah, you little chunky boy.
Here's a rapper. He'll come running. Oh.
Yeah. Hey, you hear they have tasty treats? All right. So the DNA, so we can turn that RNA from the HIV into DNA and then do PCR using primers that look for genes that are associated with HIV. And so remember, with PCR, we have 3 steps. The first one is denaturation. The second one is anneal. And what are we anneal? What does annealing mean anyways? Or binding things back together. So that's what anneal means. Bringing things back together, binding them together. And so annealing, we were going to anneal our HIV primers. And then third step is going to be elongation. You'll only be able to elongate, add nucleotides if the primer is bound, because the nucleotides that we're adding on to match the A to the T and the G's to the C's and everything like that, those nucleotides that we're adding on can only add on to something that already exists. So if the primer's bound, then we can add on the nucleotides that match. And that's only going to happen if you're positive. So that's a way we can test for the genome of the HIV. If somebody has a viral load that we say is undetectable, so 0, okay, even that so he could lay on it, but I guess he'd just rather be cuddled on, whatever. So he is really a baby. Anyway, so if somebody has an undetectable viral load, their actual HIV virus in their blood is low enough that our machines said there was none in there. So if we do this genome test on somebody that's been on antiretrovirals, on those same drugs that we used to treat HIV with, it's the same ones that we use for PrEP and STEP and things like that. So if somebody has HIV and they're on an antiretroviral drug and their viral load is 0, what will they show up with on our genome test here on our PCR? They'll be negative. They'll be negative on our PCR test. Okay, so there's that. So another thing we can do is look for the actual HIV antigen. Now, not looking for the antibodies to it yet. We'll come back to that, okay? So right now we're talking about looking for the HIV surface antigen, maybe the spike proteins. So what we will do is use a known anti-HIV antibody and then do ELISA asking the question, does the patient have HIV antigen? What type of ELISA is that? The sandwich one, yes. So that one is direct. If somebody has an undetectable viral load, somebody that we would say has zero virus in their bloodstream, what would they test for if we were looking for the antigen? Would they be positive or negative? They'd be negative. If they have an undetectable viral load. They still don't have virus. We still can't see the antigen. Okay. So how do we actually look to see if somebody had HIV at any point? That's when we come up with testing for the antibody. Okay. But to test for the HIV antibody, we'll do ELISA again. But this time in the ELISA, we're going to ask the question, does the patient have any anti-HIV antibody? This is a more important question because if you've been on antiretrovirals and your viral load is zero, undetectable, you could still have antibody in your body because all the way back up to the very, very top where we first saw the outside infection, where we first saw, there's too many greens. Let's get this. Where we first saw outside HIV, and then we activated our B cells and made plasma cells, and then we made IgM and IgG. From that, if you have had that reaction, you have had HIV in your body. So you must have had HIV. So asking if our patient has anti-HIV antibody. And so what kind of ELISA would that one be? Indirect. Oh yeah, they want to see. I just saw they wanted to see that. Let me see if I can turn it on just briefly for you guys. We'll take a little mini break. You guys can see my husband holding it all delicately over here. There it goes. Let's see if I can. I'll just show it this way. There's Meatball and my husband. This is Brad. Hi, Meatball. You good boy. Yes, he is. Okay, so now you guys can't say you didn't get to see Meatball. Yes. Yes, it's on Zoom. I'm sure this is going to pop up right. All right.
Similar with hepatitis, like
Yes.
B or C.
So hepatitis B and C both incorporate into the DNA of the host. And so we will look for antibodies for them as opposed to the virus. Now, most of the time when you have hep B or C, you'll also have a viral load with those guys. Yeah. Huh? What?
He's not chunky, he's big boned.
Yeah. Yeah. Trust me, he would be chunky if we let him do what he wants to do because he'll just eat anything all the time. You're not looking, and suddenly he has found a way to eat something that you don't. I will get food, guys. I'll get food and I'll have it out on my table, and I'm like, oh, I need to go grab a drink. I'll be like 20 feet away, and I'll look over, and he's already like wolfed down half of it. Yeah. All right, so we've got our indirect ELISA, and now we can look to see if we get that color change with our ELISA. That's basically driven by our enzyme interacting with the substrate, and the enzyme's only going to be there if the secondary antibody made it that far, and the secondary antibody only makes it if the antibody from the patient was there. So that's our thing for that. Yes. Okay, that's good. I have a whole page dedicated to just basically saying that. That's great. All right, so moving on. So that pretty much is everybody following what's going on with HIV and our CD4 T cells. Does anybody have any questions about that anyways, like that you can think of off the top of your heads before I move on to the next thing? Okay, so we're going to move on to a staph infection now. Now this person has gotten a staph skin infection. Let's say it's somebody that was shaving their face or their legs, and then Staphylococcus aureus got into the body that way. All right, so here now we will have, we already know if we're talking about staph skin infection, and if I were to draw them like this, is that representative of what you would expect to see with Staphylococcus aureus? Round cells that are arranged in clusters. Yes, so we know this already. If it's Staphylococcus aureus, staphylo is the irregular bunching. Clusters. And coccus is the round shape. We have now gotten Staphylococcus aureus introduced into our body. It's not inside of the cells. It's still an outside infection. I'm not going to go over the immune response for this one directly just yet, okay? So this is an outside, okay, for sure I can spell. Outside infection. We'll come back to this in a little bit. All right. So what happens is, let's say you cut yourself, the bacteria are getting into the body, and now these cells are going to try to basically make copies of themselves. Okay, some other things, right? So some other things that we're going to say about this. Staphylococcus aureus. We know Staphylococcus aureus is gram positive, right? If we were to do a gram stain on it, what color would it be? Purple. All right. What about endospores? Does it make endospores? Right, negative for endospores. And why do we know that? Because the only species that make endospores are Bacillus and the Clostridium bacteria, right? All right, so that's the next one that we'll get to, right? Is Staphylococcus aureus, is it acid fast? No. This one is the one where you're only acid fast if you're Mycobacterium. So it'll be negative for the endospore test. It'll be negative for the acid fast. If we were to grow this on mannitol salts agar, I know this is lab stuff, but we're bringing it back, okay? So mannitol salts, what was our selective agent? So this is just regular salt this time. Yeah, it's okay. The differential was whether or not it fermented mannitol. That was another one that a lot of people got wrong on the lab test, because I asked the question on that media. It asked what would happen if an organism fermented lactose on this media. That media doesn't test for lactose fermentation. And that was the answer to that question. So FYI, you guys. All right, so mannitol salts, whether or not it ferments mannitol. And if it does, it'll turn it yellow. This is a characteristic of staph. All right, so we kind of brought some stuff back there. Let's see where I wanted to lead next. Yes, we are going to talk a little bit about the outside response as well as the bacteria when it gets into the body. Okay, so if it's an outside response, then an antigen presenting cell, like a macrophage or something like that, macrophage, dendritic cell, whatever, will come and eat this up through that process of phagocytosis and take it inside. And because it came from outside, it'll be expressed on MHC2. We already know this. Here's some staph antigen on the surface. This will be recognized by a CD4 positive T cell. Right, we're coming back to this, and we've gone over this enough that I'm not going to go over all the details again. But this is CD4 that recognizes that. These guys will send some activation signals, causing this to proliferate and differentiate and become Th1 and Th2 cells. The Th2 cells will then go on to activate our B cells that also see that antigen. And so these guys will turn into plasma. And the plasma cells will crank out IgM first, and then class switch into IgG and whatever else. Right. So that's our immune response to what's going on in here. Just to summarize it, just say we went through it, right? And we don't need an inside response. We're not going to need to see a response of our Th1 here. We don't have inside stuff going on. So we're not going to get a lot of signaling on that side, but we'll get a lot more signaling on our outside captain, the Th2 guy. Yeah, meaty, meat. Okay, so with the bacteria, this is on our side of it, but with the bacteria. Now they're in our body. And now let's say, so here's another thing about staph. So this is our staph cell. It's got a little genome in it. It's going to be a circle. So actually, let me draw it that way. It's got a circle DNA and whatever. Here's this little DNA in a big old circle. I'll flash like this for a little bit, just bear with me. So that you know that that's its DNA genome. It'll have ribosomes and whatever crap else out here. And this is its inner membrane, or the cytoplasmic membrane. All right. So if Staphylococcus aureus, is gram positive, is it going to have a thick or a thin cell wall? Thick. And what's that cell wall made out of? Peptidoglycan. Excellent. So here is, actually wish that I had changed that color. Let's see if I can. All right. Yeah. All right. So this is our wall for our gram positive. And Staphylococcus aureus also has a capsule around it. Whatever. So what'll happen is if this was your body, come on thing. If this was your cells here, maybe in your skin, so maybe your dermis cells, that these guys can now stick. Capsule allows for adhesion here. But what else is the capsule going to do? The capsule helps evade phagocytosis. So now up here where our macrophage was, this part here, this part, why is this so thick? This part right here where we're having our macrophage phagocytose things, this might not happen because it might be avoided because we have that capsule. It makes it hard to eat it up. We might eat up some of them. And if we can eat up some of them, then that'll give us enough drive to be able to make antibody down here. All right. So the antibody is important. How is that antibody going to help against staph? All right. So first things first. The classical pathway or complement activation. This has several functions in and of itself. So remember, there's 3 ways you can activate the complement system in your bloodstream. Either because you saw mannose on the surface of bacteria, using the lectin proteins in your blood, and then the complement recognizes that. Or it might be that there's LPS or peptidoglycan or something like that, that can trigger the alternative pathway of this. But the classical pathway happens whenever there's antibody bound to something. So if we have our bacteria down here, here's our bacteria, and it has a little capsule on it. As the wall too. Okay, fine. We can draw the wall again. Here's the wall. And then was our capsule. And so it might be able to evade phagocytosis like this. But now we get antibody that binds onto that. Maybe this is IgG. And now this help with the phagocytosis being able to grab it. So it acts as, this can act as a handle. If we're talking about this in the sense of complement though, this will act in a couple ways. So this is what's going on with our cells. So I'm gonna bring this out to the side because I want to come back to this in a little bit and how our antibody is gonna be worked on. So I'm put this down here for reference. This is where our antibody is kind of doing. So when it activates complement, what's going on is we can have complement seize the antibody and binds to the antibody to help act as a molecular handle. That will help phagocytosis. What's the term for this? Anybody remember? Anybody remember what this term is? It's opsonization. All right. It can also complement, can also just initiate the inflammatory reaction. So this can already be happening just by seeing peptidoglycan from our PAMPs there, but it can also be happening because of our activation of our complement system. And so if our antibody comes down here and binds onto our cell, we can now start the inflammatory process even more. We can help with phagocytosis. Is he trying to pee?
I don't.
Is it a bug? Oh, a fruit snack. Yeah. There's no trash cans in here. Just leave it up there. Yeah, I'll take care of it. Buddy. So anyways, and then also there's another thing that complement can do, which is the biggest thing that complement can do, which is activate the membrane attack complex. And this will poke holes in the bacteria, then killing it. So already antibodies doing that, but there's some other things that antibodies going to do as well. It could coat. So blocking the bacteria from attaching, blocking it from interacting with other bacteria. It could cause agglutination, causing the bacteria to stick together in big clumps. All right. And there is another thing that it could do, which is just direct helping with opsonization. So the antibody can be the handle, or the complement interacting with the antibody can be the handle, either way. All right, so this is how our immune system can help dealing with our outside pathogens by having all this. Now this will happen, this outside stuff with our, activating the complement pathway and coating and agglutination. This would happen to the outside HIV as well. So if we have antibody to HIV, all of this stuff is happening with HIV, no matter what it is. If you have antibody being made, it could be acting in these ways here. This is just giving an example with a staph infection in our body and how we can deal with that capsule by helping with opsonization and that sort of stuff there. All right, moving on to a different scenario. we're gonna have a scenario where there's an outbreak at the state fair. So at the state fair, over the course of a week, all right, over the course of a week, we will have 700 people get diarrhea. Not dysentery, it'll just be regular diarrhea. Okay. So it's been over, like this has been happening. There's been several cases over the span of a week. And so maybe as an epidemiologist, going and visiting people, honey? Yeah. Are you a meaty boy? Meaty ball. You're so cute. Mama's guy. All right. So in the epidemiology of this, if you were the epidemiologist here, maybe you'll gather a group of people who have the disease, which we know there are 700 of them. And so, well, maybe we'll find 700 people that are uninfected. And now we'll look at where they ate when they were at the fair. Okay, so maybe they could have ate at the hot dog stand, the funnel cake place, maybe the fried chicken stand, maybe Indian tacos. So here's the data that we collected. Meatball, where did he go? Okay, okay. All right. So here's the data that we collected. Of the people that were infected, maybe 100 went to the hot dog stand, maybe, I don't know, 150 went to the funnel cake place, but all 700 of them went to the fried chicken place. And maybe some of those also went to the Indian taco place. I don't know. It doesn't have to add up to 700 because they maybe went to multiple places. If we are looking at uninfected people, none of them went to the fried chicken place. And maybe we had, I don't know, 500 of them go to the funnel cake place, 100 of them went to the hot dogs, and 100 of them went to the Indian tacos. But none of them went there. So now our attention is being drawn to the fried chicken place. So with this information in mind, what type of epidemiological study did we do? This is way back, guys. Does anybody remember? So I'll give you, I'll give you some choices here. All right. Is this, so we have case control, cross-sectional, or we have cohort. Those were the three studies that we talked about. Anybody have a guess as to which one this is going to be? And there's not very many of us, but all right. So this is going to be the case control study because we're looking at cases and we're looking at the control group. So the control group is the uninfected group and the case group is the infected group. So the ones with the cases. All right. So this is a case control study. A cross-sectional study would be if we just jumped in and we said, okay, everybody that's in here, okay, do you have the disease or not? And what did you eat and what didn't you eat? But looking at that cross-section in that moment. But instead, we're going and taking everybody that got sick and comparing them to a good group of people that didn't. So the cases versus the control. All right. So that's the first things first. Second things is going to be what type of epidemic is this? Type of outbreak. There's a couple of things. Is this an endemic situation? No, because this isn't something that occurs at this level all the time in the area, okay? It's not an endemic. So this might be an epidemic, but it also might just be sporadic. If I needed to know the difference on the test, I'd give you more information in that regard, okay? It might be epidemic. Is it pandemic? Why not? Right, we didn't cross continents, exactly. And so another thing we can know about the type of outbreak, we can even look at it this way. What am I trying to get to? I'm getting distracted because you got other things going on. Okay, sporadic source. Okay, right. So for the source, there were different kinds of sources that we could have from where our epidemics stem from. Is this a point source, a common source, or is it a propagated? All right, that's all right. So it's not point source, right? Because if I were to sneeze in here and you guys got COVID, that was one incident. We can narrow it down to one place, one time that all the people got sick from. This is happening over a week. So it's not necessarily from one piece of chicken that got people sick, right? This is basically probably how they're handling their stuff. So probably more likely they're frying up their chicken, and it could even be cooked, but they're probably replacing it in the area where they had the raw chicken or something like that. So this is happening over a greater period of time. This is not a point source epidemic. Propagated epidemics are like what we have now with COVID. The human population is keeping it going. So it's propagating it within the human population. And so that's what we would see with COVID. So this is a common source epidemic. And our example that we had in class was whenever John Snow discovered the outbreak of cholera and linked it to that water pump in London. But people were getting sick over a long period of time, but they were all visiting that water pump over a long period of time. So this is a common source. It's all coming from a common source, which in this case is our fried chicken stand over a long period of time. So this is a common source epidemic. Now let's look at what we think is causing the disease. So we think people are getting this from chicken, fried chicken. What do you think is the pathogen? More likely than not. You have any guesses? Salmonella. So salmonella is also known as, the species name is Salmonella enterica. Something that we also know about salmonella. Salmonella is a motile. It's actually peritrichous. What does peritrichous mean? Right, so if this was our salmonella, it's a rod shape and it's got flagella all over it, many flagella. So we could actually look at it with a flagellar stain and see all that flagella and identify it that way. Flagella all over it. All right, it's also gram negative. And as it says in its name, it's a gram negative enteric. Can you guys think of any of the media that we used in the lab that was specific to gram negative enterics? Maybe it had bile salts in it. So it's okay. You're close. It's one of the red ones. You're right about that. So this is McConkey. So you won't need to know this for the lecture, but tying this back to what we learned in lab, right? I know we're done with lab, but this is something that we could do to grow a gram negative enteric. All right, so that's something we could do. We could also do a gram stain. What color would it be on the gram stain if it's gram negative? Red. So we could look for motility. We could look for it growing on McConkey salts. We could do a gram stain. So this is some stuff that could tell us about it. One of the things that we learned in lecture is that we can identify some of these enteric bacteria by their HOK antigens, which has to do with their capsules, their flagella, and other markers on the surface of the cells. So if we're looking for antigens specifically, then we will usually use antibodies to identify. So if we're using antibodies to identify that, this would be an immunological test. If we're looking for the antigens using a known antibody, that would be direct for that one. All right. If we're doing one of these tests up here, this isn't an immunologic test. We're growing to see if it grows on McConkey. If we're doing a gram stain or whatever, this isn't going to be immunological, right? This isn't genetic either. So the term for this type of test would be phenotypic. You guys remember this stuff? Another thing we could do to identify this guy would be to use PCR with salmonella primers. Salmonella gene primers. If we do this, this is a genotypic. So we've got tests in all of our three major categories that we could do for this. In order to do these tests, though, the first things that we would need to do is go through the 5 I's, where we would inoculate, incubate, isolate, inspect, and then identify. So our inoculate, we would take maybe a fecal sample or swab of where the chicken was, and then grow it on some media there. So we'll say the swab of the chicken area, or patient fecal sample. Then we will incubate it at probably around body temperature. Then once we have that sample growing up, let's say you got a fecal sample, you'll just put it in some broth, let it grow. Now we can take that and streak it out on like a McConkey agar plate and kill 2 birds with 1 stone. Let's grow it on McConkey agar. It should be able to grow on it if it's an enteric bacteria, right? And we can prevent any gram positives from growing or any non-enteric contaminating bacteria from growing, and also do our quad streak method to get our individual colonies. So we can take an individual colony and create pure cultures that we can do our studies on. So quad streak. Now we can do our inspect test, or we can do any of these things up here, PCR, HOK antigens, etc. And then use this data to identify it as salmonella. Okay, so that's the process that would be involved in going from people are sick to they have salmonella. All right. We did our epidemiological study. We did a case control study to notice that this was a common source epidemic or sporadic outbreak. All righty. So for our treatments for this, this is, again, Salmonella enterica, which is gram negative. All right. So this is a bacteria, right? What drugs do we use to treat bacterial infections? Antibiotics. So we have several groups of antibiotics, the ones that attack the cell wall, the ones that affect the membrane, ones that affect folic acid synthesis, ones that affect nucleic acid synthesis, and ones that affect protein synthesis, or the ribosomes themselves. We're going to go through and give some examples of each of these. What's an example of a cell antibiotic that affects the cell wall? Penicillin, all of our cillins. Some other ones might be cephalosporin, carbapenem, and vancomycin, and bacitracin. Anybody know any of the membrane ones? Polymyxins and daptomycin. Folic acid synthesis inhibitors. These are our sulfas. All right, we could go through them, but you'll probably be able to recognize them by now. Nucleic acid ones. These are the group is known as the fluoroquinolones. Examples, they're all going to end in floxacin. So what's an example of that one? Cipro. All right. And our antibiotics that affect the proteins synthesis as well as ribosomes. Many of these will be the other ones that end in mycin, but some better examples might be something like tetracycline, glycylcycline, and then we have like erythromycin and streptomycin and that sort of stuff. All right, so we've got some examples down here. If this guy is gram negative, he has a thin wall, right? And an outer membrane. Would you prefer to use a cell wall affecting one, or would you want to pick something else? So I'd probably go with something else, right? This is because the gram positives are more strongly affected by our cell wall inhibitors. So I'd probably reach for something else. And just from this alone, we wouldn't know necessarily what to pick for, but we could at least narrow it down to probably not the cell wall inhibitor guys, right? Use them at high enough concentrations, they might still be effective. They might be safer still. So if you have a patient that has mild diarrhea, but they're not getting over it on their own, you might give them a higher concentration of one of these cell wall inhibitors, and it still might work. But you could also reach for one of these other drugs. The problem with these other drugs is that they might also have their own toxicities that could be affecting the patient. But yeah. All right. Any questions about this stuff? All right. We're going to go on to operons. Man, has it been a while. You guys remember bacterial operons? Right. So an operon is a set of genes all regulated by the same promoter and operator. All right. So I want to make sure that I'm approaching this correctly. Make sure I'm covering all my stuff. Oh yeah, we talked about these trophes and stuff like this. If you guys want to cover that, we can. But I'm gonna post all the notes from all my classes once I get done with everybody, so that everybody has all the notes and all the videos from all the classes. So if there's something else that we talk about in another class, you would be able to go back and see that stuff. But for our bacterial operons, it's a DNA, in the actual DNA, a set of genes, all right, that are regulated by the same promoter and operator. All right, so let's look at these guys. So when they are on versus when they are off, always, no matter what kind of operon we're talking about, this is what it'll look like. So we have the promoter region, and a promoter, does anybody remember what a promoter is? This is where RNA polymerase binds. Binds on the DNA. Move this guy down and give us a little space to write this. We have the promoter, we have the operator, and then we have our genes. Gene 1, gene 2, whatever, gene 3. And they are all linked together. All right. I wish I'd made a copy of that before let's try this. Don't you love it? Okay, so we have RNA polymerase. Make it bigger. Would bind right here and then be able to go through here and transcribe the DNA into the RNA. So what is actually happening when we do that? We are taking our DNA, which you guys know how this is, right? And we're opening it up. And if your DNA was like this, and this was A, T, A, C, C, G, T, and there's the opposite side of it, whatever, and we would write that out, it would be complementary to that. If we're copying on this side, our RNA polymerase would come in and go from 5' to 3', and it would make RNA that would match up to it. So this would be U, A, U, G, G, C, A. And this would continue on until we made our whole RNA and we got to a terminator sequence. Then our RNA polymerase would fall off. And so would the RNA, and we'd have our single-stranded messenger RNA that can then be translated by our ribosomes, right? Matching up our codons with our anticodons of our transfer RNAs, carrying our amino acids, linking the next amino acid to the next one with our peptide bonds and creating a polypeptide or functional protein even as a result for each of these genes. All right, so this is what's really happening with our RNA polymerase. So what happens is when you have, there we go, okay. When you have operon that is being turned on or off. Let me show you this, highlight. Whenever RNA polymerase is here, and then our, this thing is here, this thing here that I'm drawing here is called a repressor. So now, when RNA polymerase tries to go, it won't be able to, it'll be blocked. All right, so inducible operons, they will start off. All right, so they start in this one. So inducible starts here. We have the repressor on there. Once we have a molecule come in, which we call the inducer, which might be lactose, which we talked about in the lac operon. Repressor. So what are you doing?
He went under the chairs.
Oh, that's okay.
Like,
The inducer binds the repressor, so it'll come off. And that'll turn the operon on. In repressible operons, this is really big. I'll make this smaller. They will always start on. Then whatever product they're making will bind with another protein to make the repressor, which then binds at the promoter, sorry, the operator, to block the RNA polymerase and thus turn it off. So it's just a matter of whether you start on or off. But either way, if you are off, then your repressor is bound and it blocks the polymerase from moving. So if you are an inducible operon, you are on. Then, sorry, if you're, no, I'm going to just back that up. If you're inducible, the only way you can be induced is because you are off, right? Just like if you're in labor, the reason we induce you is because you're not in labor yet, and you should be. So that's what we're going to be doing. We're going to turn you on, but we need to turn you on because you're off. So an inducible operon, it is turned off. Then something comes in, binds with a protein that can then remove the repressor, and now it can be turned on. If you're a repressible operon, then you are on. You're making too much product. The cell isn't using it up. It gets built up, just like if you were a business selling, dresses. Too many dresses are made. You're going to stop making the dresses. Same thing with the bacteria. We're going to stop making this product for the cell. It's not using it up. And so that product, the dress in this instant, we get too many of them. They get brought up to the repressor protein, and then that causes it to bind on and block making the genes that are associated with that. So this is only going to be in bacteria that they have this because they have these sets of genes. For eukaryotes like us, we have each gene has to be regulated individually. All right. I know it's a lot. Do you guys have any questions about any of this? Do you want me to go over anything else in our time together before we go? You guys online? Miranda, do you have any other questions that you want me to go over? I will have one more STEM lab day. So on Monday from 2:30 to 4:30, if you guys need to review anything, I'll be available in the STEM lab on Monday from 2:30 to 4:30 if you want to come by. That's my last day in the STEM lab. Because I know you guys have your final exam on Tuesday, right? So you guys do still have that opportunity, and you can always email me if you have questions, or if you watch any of these videos or look at any of these notes and there's something you're not understanding, you can always email me or set up a Zoom with me, and I'll be happy to meet with you guys to help clarify this stuff. Otherwise, I think we're done. Yeah? All right. Yeah. See you on Monday, Miranda. And you guys are welcome to come as well. Yeah, that's it. We'll go ahead and end our review, and I will get this posted and the video with this posted for you guys to review when you're studying with your stuff. Remember that tomorrow I have, if you guys watch the videos or anything, it doesn't matter, but tomorrow I'll be doing my weekly review for week 16. I'm just going to talk about chapter 24, so it should be a pretty brief one. And then on Sunday, we'll play through the Kahoot games. Then Monday, I'll be available in the STEM lab for you guys. Yep. All right. Good luck studying.